therapy of bromodomain inhibitors with HDAC inhibitors in neuroblastoma, which shown synergistic anti-cancer effects through suppressing a couple of oncogenes. So neuroblastoma is the most common childhood solid tumor, which is developed in the nervous system outside of the brain, with an average age of diagnosis of 18 months. And it accounts for 15 to 20% of childhood cancer deaths. Usually the patients are categorized into 5 different stages based on patient age at time of diagnosis, tumor biology, and MYC amplification. In stages 1 or 2, or which are considered low risk, tumor is usually localized in the spinal cord or adrenal glands, which can be easily removed with surgery alone. Whereas in high-risk group, tumor is spread throughout various locations in the body, such as lymph nodes and bone marrow. And usually, despite intensive chemotherapies, these patients undergo relapse. And more importantly, this class of patients are also strongly associated with high MYC amplification. So MYC is an oncogene which functions as a transcription factor. It's expressed in several cancers, and there's more than 200 copies of MYC found in neuroblastoma. And 25% of neuroblastoma is actually caused as a result of MYC oncogene amplification. And usually the patients with high MYC amplifications have a poor overall survival outcome compared to no MYC amplification. Basically MYC functions as a master regulatory protein in cancer. So it's a protein that's synthesized by the cell, which activates the expression of other genes, almost like a light switch. So MYC doesn't function on its own. It binds with another molecule, MAX, and wherever they sit throughout the nucleus on the DNA, they activate the expression of the genes in that region. Evolutionary, MYC binding site is positioned adjacent to the genes that control growth. And physiologically, we need MYC in the body to drive the formation of new blood cells. However, cancer steals MYC, maintains it in one position, and therefore makes the cells to forget to stop growing. So today, therapeutic strategies to target MYC simply doesn't exist, mainly because the structural analysis of MYC protein has failed to identify a hydrophobic pocket into which an inhibitory molecule could be designed. But recently, quite a few studies have shown the important role of bromodomain proteins, particularly BRD4, in modulating MYC transcription activity. So the proposed model is that bromodomain proteins such as BRD4 recognize the acetylase residues on the histone and then bring the whole transcription machinery complex at the promoter region of the MYC target genes and therefore activate the expression of the growth-related genes. And on the other hand, bromodomain inhibitors, they remove BRD4 or bromodomain proteins from the chromatin, therefore disrupting the transcription machinery complex and shutting down the activation of the gene expression. So bromodomain inhibitors are small molecules. They downregulate MYC and MYC target genes, and so far has shown promising anti-cancer effects in several cancers. And one of the latest versions of bromodomain inhibitors, IBET-762, is actually in a clinical trial right now for the treatment of leukemia and lymphoma. So first, we tested a panel of MYC-amplified neuroblastoma cells with JQ1, and the results showed a dose-dependent reduction in the number of viable cells up to 1 macromolar of JQ1 in almost all the cell lines. However, increasing the dose of JQ1 up to 4 macromolar doesn't have any further significant reduction in the number of cell viability. Next, we looked at MYC transcription, changes in MYC transcription level after treatment with JQ1. And these immunoblots here just show that JQ1 is able to reduce the MYC protein expression in a dose-dependent manner after 48 hours of treatment. Also, MYC mRNA level is significantly reduced after treatment with JQ1, but it's not dose-dependent. Even though our study and many other studies have shown promising effects by bromodomain inhibitors, no study yet has shown complete cancer remission with the use of bromodomain inhibitors alone. Our histone modification group in Children's Cancer Institute has been focusing on finding novel pathways between HDAC molecules and MYC oncogenes, and so far we've found quite a few interesting pathways between HDAC1, HDAC2, HDAC5 CEP molecules and how they modulate MYC transcription. Therefore, we decided to do a combination therapy of bromodomain inhibitors with HDAC inhibitors, and we chose to work with panobinostat, mainly because it's a very highly potent HDAC inhibitor that works at nanomolar concentration, which is clinically achievable, and has so far shown minimum toxicity on normal cells. And more importantly, just a couple of months ago, panobinostat passed FDA approval to be used in clinics for cancer patients. Then we tested with several different doses of JQ1 in combination with panobinostat, and the results here just show that the yellow line is the JQ1 on its own, whereas the blue and the red and the black ones here, they are in combination with panobinostat. And as you can see, combining panobinostat with JQ1 reduces the cell viability a lot further than JQ1 on its own, which is confirmed in 2 neuroblastoma MYC amplified cell lines. In a different experiment, we kept panobinostat concentration at 10 nanomolar and then combined it with different doses of JQ1 up to 32 nanomolar. As you can see, increasing the concentration of JQ1 even up to 32 doesn't really have significant effect, doesn't really kill more than 50% of the neuroblastoma cells, and it kind of has a plateau effect. But combining that with panobinostat, as shown with these yellow bars, has a very dose-dependent effect on reducing neuroblastoma cell viability. And here it just shows us that the hyperacetylation that's caused by panobinostat in these neuroblastoma cells makes these cells, increases their dependency on bromodomain protein-regulated transcription, and therefore these cells become a lot more sensitive to bromodomain inhibitors. And we also did some synergistic analysis in calculating the CI values, which is combination index value. That value, CI values below 1 indicates synergy, whereas above 1 indicates antagonist effect. And as you can see, on almost 90% of our combined doses, we've observed a strong synergy between these two drugs. Next, we wanted to know what's actually happening in the mechanism between these two drugs, and so we did a macroarray gene expression analysis. And here, this heat map is just showing that like over 2,700 genes which were modulated by this treatment. And as you can see, the combination therapy, the combination treatment significantly enhances the effect that's already caused by either JQ1 or panobinostat. It's almost like when you combine these two drugs, they enhance each other's effects. And the number of the genes that were upregulated or downregulated by fold change greater than 2 is a lot higher in the combination compared to single therapy on their own. Gene set enrichment analysis also showed significant downregulation of HDAC and MYC target genes as well. We then looked at the top 30 genes that were most significantly, most dramatically modulated by this combination therapy compared to single therapies. And then quite a few genes popped up, which interestingly BCL2 and anti-apoptotic gene was significantly downregulated. But LIN28B stood out for us. So LIN28B is an RNA binding protein which binds the LET7 mRNAs and increases MYC protein expression. And overexpression of LIN28B in neural crest in mice actually causes neuroblastoma. This figure here is taken from the TCGA website, and it just shows that LIN28B is highly amplified in neuroblastoma patients samples compared to all the other tumors. And high levels of LIN28B is associated with poor overall survival outcome. We also looked at publicly available COPEC database and found a positive correlation between LIN28B and MYC expression in patient tumor samples. So we then looked at, we then performed a Western blot analysis to look at the changes in LIN28B expression when you treat them with the same combination therapy. As you can see, panobinostat has almost no effect on reducing BCL2, N-Myc, or LIN28B expression at all, whereas JQ1 treatment significantly reduces the expression of all these three proteins, whereas combining JQ1 with panobinostat has a great impact on downregulating LIN28B, BCL2, and N-Myc expression at a synergistic level. It was also important for us to look at the apoptotic cells and to know whether these reduction in cell viability is as a result of apoptosis or just cell death. So we performed an annexin 5 staining and subjected to flow cytometry analysis. So with JQ1 or panobinostat treatment alone, we observe up to 20% of apoptotic population, whereas in combination, there is almost 65 or 50% of the cells have shifted into apoptotic phase. It was also important to look at the effect of this combination treatment in normal fibroblast cells. So we performed the same apoptosis assay. As you can see, there's negligible apoptotic population in normal fibroblast cells, so indicating that this combination therapy is not toxic to normal cells. We then moved on to animal studies where we xenografted neuroblastoma MYC-amplified cells into nude mice, and once the tumor developed to a certain size, then we treated them with either control or the JQ1, panobinostat, or the combination treatments together. And the results is just showing that JQ1 or panobinostat are able to slow the tumor progression by up to 40%, whereas the combination of the two almost results in regression of the tumor up to day 12. However, after day 14, the tumor is starting to grow again. But we couldn't continue this study any longer because the drugs were quite expensive. So maybe it's worth repeating the study for a longer period of time. We also looked at the MYC protein expression in the tumor tissues that we collected from these mice that were treated. And here's a quantitative analysis just showing that panobinostat absolutely had no effect on MYC protein expression. And though JQ1 significantly reduced the MYC protein expression in these tumor tissues, but a combination therapy had a stronger effect on downregulating MYC protein expression. Then the next question for us was to just investigate the relationship between bromodomain proteins and LIN28B, because LIN28B is an oncogene in neuroblastoma, and we were curious to see if there is a link between LIN28B and bromodomain proteins. Therefore, we knocked down bromodomain proteins, BRD3 and BRD4, and then looked at the changes in LIN28B expression. And we found that when BRD3 or BRD4 are knocked down, LIN28B mRNA expression is significantly reduced. Also, LIN28B protein expression is reduced. Therefore, we could say that bromodomain proteins, BRD3 and BRD4, they're able to modulate LIN28B gene expression. We also performed cumulative immunoprecipitation assay, where we looked at the enrichment of BRD proteins at LIN28B, various regions of LIN28B, and we found that once you treat these cells with JQ1, then bromodomain protein, BRD3 or BRD4 antibodies are no longer able to bind at LIN28B promoter regions, therefore indicating that bromodomain proteins, they bind directly at LIN28B promoter region, especially at transcription start site and the intron region, and that's how they modulate LIN28B activity. And this was further confirmed by luciferase assay, where we showed reduced LIN28B promoter activity upon BRD3 or BRD4 knockdown. So with all this, we've confirmed that combination therapy of bromodomain inhibitor JQ1 with hashtag inhibitor panobinostat is able to significantly prolong neuroblastoma tumor progression, and it also reduces MYC protein expression in neuroblastoma-bearing mice. And we've also shown a novel mechanism here of how bromodomain proteins, particularly BRD3 and BRD4, are able to modulate MYC expression indirectly through modulating LIN28B expression. And with that, I'd like to thank my supervisor, Dr. Tawalio, and Professor Richard Locke, and everybody else at Children's Cancer Institute, Histone Modification Group, and the School of Technology and Biomedical Sciences.
Thank you for the interesting presentation. Any questions from the audience? Yeah.
You have used hashtag inhibitors in your study for the N-Myc expression.
Yeah.
So, are there any epigenetic modifications for N-Myc
Okay, any more questions? Yes, please.
So in the combination therapy in your mice experiment, when you started giving the treatment, what day?
We had to wait for the tumor to reach a certain size of 0.3 cubic meters. So it took almost 2 to 3 weeks before we could start the treatment.
Okay, any more questions? Maybe I got one question for you, right? You mentioned that in your data, the combination therapy is not cytotoxic to the normal cell using the fibroblasts. Have you used the counterpart, like for example, primary neurons as a control?
No, we
Okay.
haven't. We only tried 2 different normal fibroblast cells, WI38 and MRC5 cells. So those showed no
Yeah.
toxic effect.
Because it's quite interesting, you work on the neuroblastoma, you can see the specificity, it's a high potency. And the other question is about the ability of the combination therapy to induce neuronal cell differentiation. Have you looked at the differentiation of the neuroblastoma cells?
Not in this study, no, we didn't look at that one in this study.
Okay, thank you.